Well, good afternoon, everyone, and welcome to the front row. My name is Jamie Williamson. I'm a professor here at Scripps, and let me be the first to welcome you. We have quite a few people here in attendance, and we've got another big group that's coming in virtually online, so welcome to everybody. And today we get to welcome Danielle Grotjahn. And I want to say a little bit about, by way of introduction, about what she's working on. But she's working on mitochondria and trying to understand their role in disease. And she's using some very powerful and cutting-edge and evolving technology called electron tomography. So mitochondria are particularly important in our bodies because they are the powerhouse of the cell. So they're actually kind of a furnace. Their job is to be this little compartment where you turn glucose into ATP. And they try to do that as efficiently as possible, but some amount of that is released as heat. And so the fact that we maintain our body temperature is in part due to this throwing off of excess heat. So they're really not only fundamental to the function of the cell, but to us as an organism. And I'm going to let her explain the many ways that the dysfunction of mitochondria might be involved in some very common diseases. Now, I want to say a couple of other things. First of all, it's our 100th anniversary. So, yeah. Ellen Browning Scripps founded a set of institutes 100 years ago, and we are the descendant of one of those. And we have a lot of sort of centennial activities that are going on, so stay tuned. But it's just a great time to reflect on how did we get here and where are we going. So it's been good to do that. Now, I want to say something about the Fellows Program. So Danielle was one of our first fellows. We have this program called Scripps Fellows. And we patterned it after a couple of other successful programs. You might have heard of Whitehead Fellows at MIT, Miller Fellows at Berkeley, but we have Scripps Fellows here. But the idea is that we capture someone right out of graduate school. Typically, you get your PhD, and then you go do postdoctoral study somewhere, and then apply for a faculty position. So what we do is create this fellow position, somebody right out of graduate school with tremendous promise and potential, and we give them this mentored environment to develop their independent program. And at the end of that, so this is a 3-year position, at the end of that, we want to hire them onto the faculty. So I forget what number you were, number 5? Number 3. And so we've had about 9 or 10 fellows, and I think 8 have actually joined the faculty. So it's been a tremendous way to get people at the cutting edge and coming out of fantastic labs with new things. They bring it to Scripps. And Danielle is the one that really set up cryo-ET here, cryo-electron tomography here at Scripps. So it's a great program, and she's off to a tremendous start. So she's one of our young assistant professors that's working on building their program. So this is very much looking at the future. This is the new thing. I'm going to retire someday, and we need the next generation to replace us. And so Danielle's off to a fantastic start, as you'll see. And then the last thing I want to say is something about basic research. So a lot of the front row lectures tend to focus on drug discovery. We're talking about curing diseases, finding new medications. And that's great, but I would say that's probably somewhere between a quarter and a third of what goes on at Scripps Research. A lot of what we do is just basic research. What does that mean? It means, how does stuff work? The things that that Danielle is going to talk about are just trying to understand mitochondrial structure and how that structure relates to its function. And sure, it's important for disease, but she's not making medicines. She's trying to figure out how stuff works. And it's really, really important that everyone understand we have to support basic research. That's the foundation on which medical discoveries are made. And without that, there can be no progress. So before Danielle comes up, I want to say just one thing about what happens at the end. So she'll give her lecture. And at any time, if you have a question, You can type it into the chat if you're virtual. If you're here, save it. And at the very end, I'll come up and Danielle and I will sit here. We'll have a little discussion. I'll get fed some questions from the virtual audience, and we'll have someone, just put your hand up and someone with a lightsaber will come so that I can see you and call on you, and we can try to have questions from the audience. Don't be shy. Ask anything you like. Probably no medical advice will be given at this particular session, but we're happy to have any kind of questions that you'd like. So I will join you at the very end. Sorry, I just said all of that stuff. So I'll join you back at the very end, and please, let's welcome Danielle.
Hello, everyone. It is my pleasure and honor to be presenting to you today at our final front row lecture of 2024. I want to first acknowledge the incredibly hard work of both Jamie and the development team for putting on this fabulous event. Let's give them a round of applause. So I'm here standing in front of you today, giving you this front row lecture, thanks to two incredible women who have really influenced my life. The first is my mom, Julie, who for the first 10 years of my life, it was just her and I. She raised me as a single mother. We had an incredibly close bond. We did almost everything together. And the second woman that's very important to both of our lives was my mom's mom, a very special lady known as Grandma Pat. And I'm biased, of course, but those that know Grandma Pat know I'm not being hyperbolic when I say she was one of the most kind, patient, and loving women to ever walk this earth. And she was the ultimate homemaker, which made for an incredible mom and grandma. And something that I really appreciate is that both of these women really enabled me to be a very curious child and allowed me to explore the natural world around me, which was quite easy because I spent a great deal of time at my grandparents' house. They had a beautiful 5-acre piece of land. I would go around exploring, playing make-believe, making all sorts of stories in my head about adventures I was going on. I would find treasures in the creek, known as Pigeon Creek, out back, and even enjoyed all of its glory in all of the seasons. And I know some of these pictures frighten us as Californians now, but I promise you as a kid, this was very wonderful to have this ability to explore the nature around me. And in retrospect, I realized that my grandma took a lot of inspiration from the natural world as well in her work as an artist. And she had several mediums in her art, but one of my favorite mediums was that she was a very talented quilter, and she made these beautiful decorative hanging quilts, which here are just a few examples. I think you can appreciate really how she took the beauty of the natural world around her that really inspired and influenced her. And this was imparted on me as well. And there was a pivotal moment for me that really set me on a trajectory that led to my scientific curiosity, which was that for my 9th birthday, I was gifted a microscope kit, a beginner's microscope kit. Now, this isn't the exact one, but it's something very similar to what I received, and I couldn't be more excited. I remember ripping open that package, setting everything up, and then performing some of my very first experiments, where I would scrape the inside of my cheek, I would smear that onto a glass slide, drop some magical dye, blue dye liquid on top, sandwich it in between a glass cover slip, and just looking at the thing, it was a little bit of a mess and it didn't look that interesting. However, when I looked at it underneath my new apparatus, I discovered an incredible new world, a world that was completely invisible to my naked eye, but really opened up my fascination for the microscopic world. And this was really from a young age where I gained an appreciation for the idea that we could think about life across scales. So what we mean by this is certain things like me standing up here in front of you, maybe my finger, maybe you can look down at a strand of your hair. Those are parts of life that we can access with our naked eye. However, there are smaller machinery that enable all facets of life that we aren't able to see with our naked eye, but we can see with the help of technology, for example, using microscopes. We can use technology that enables us to illuminate these tiny, tiny compartments that make up all of the human body. And we can use lenses that are not so different from the lenses you may have in your glasses to magnify these and discover entirely new worlds. And so this, to me, was really my journey to understanding and appreciating the microscopic world that you'll see has carried me throughout my scientific career and even to this day in my lab. And so I was motivated by this curiosity to attend undergraduate at the University of Wisconsin-Madison. Go Badgers! And the University of Wisconsin, UW-Madison, is an incredible public university in a lot of ways, but one of the really important ways for me was that it offers a lot of opportunities for undergraduates who have no experience working in a lab to work in labs that are doing cutting-edge research. And I joined the College of Agricultural and Life Sciences, and they had this really cool program called the Honors and Research Program, which enabled you to have access to these labs and kind of took you throughout all your years of undergrad through working on a research program. And so it was here that I joined the lab of Francisco Pellegri. This picture is a very old and treasured picture. You're not imagining things. It's a little bit damaged here because it's so loved that part of the picture has gotten stuck to the frame. But nonetheless, I hope you can appreciate the group, and in particular, Francisco Pellegri, who was my first mentor and really set me on a path in research. And in his lab, we used a fish called the zebrafish. They're these really tiny fish. And what we use them for is because they produce eggs that you can then fertilize that are transparent. And why this is important from a microscopist's point of view is that it gives us access to look at all the inner workings of inside of the cells that will enable them to function. And so I spent several years of my undergraduate, as well as a technician later, collecting freshly fertilized eggs and literally watching them divide, watching life happen before my eyes. So they would go from 1 cell to 2 cell. That's the time I was taking them and running up to the microscope room. And then by the time I got to the microscope, I could capture pictures of them at 4 cells, 8, 16, 32. And the goal for my project was that we had this hypothesis that the machinery that helped them divide was also dictating the overall shape as they were developing. And so I would capture these images. And I was paired up on this project with a graduate student in the lab who was visually impaired. And this was made for an incredible team between the two of us because he was very gifted with numbers and computers. And so what he taught me was the value in transforming microscopy images into numbers that we could gain meaning from. Now, why is this important? Well, quantification from these images is important because, let's face it, life and biology, it's a little bit messy. No two people are the same, no two cells are the same, but trends do exist. And so our job as microscopists and as biologists is to figure out creative ways that we can quantify what we're seeing in our images, put them into numbers so that we could run statistics that tell us what is due to, let's say, differences that might be due to a healthy versus a diseased cell versus differences that might just be due to the fact that no two cells, no two people are exactly alike. And so this was really started my journey of another important aspect that we still do in my lab, which is thinking of creative ways to quantify the images we have. And some of my lab members that are in the audience here might get a kick out of this, that this is actually something that I drew as an undergrad. And you could go on the look in my lab today on the whiteboard, and you might see very similar sort of schematics there. And so this led me to completing my honors thesis at UW-Madison. And then it came time to think about going to graduate school. And this whole time, I had been working with a special type of microscope called the light microscope. Like I mentioned before, we use light to illuminate the unseen world. But throughout my journey in undergrad, I had learned about another type of microscope, the electron microscope. And what's powerful about the electron microscope is that it enables you to look at even finer details of even smaller machinery of life. Everything down to the compartments I'm going to talk about in a little bit, mitochondria, to the viruses that can cause global pandemics, to the motors that power our cells, and even the DNA that provides the blueprint for all life. And so I knew for graduate school that I wanted to go to a top-tier school, and I wanted to learn how to do this electron microscopy. So after talking to my network a little bit and doing some Googling, of course, I arrived on Scripps Research as being one of the top places to learn this, and so this is why I decided to come here for a PhD. Now, there's lots of people that really led to Scripps being a top-tier place to do electron microscopy, but in particular, professors Clint Potter, Bridget Carragher, and Ron Milligan, they were really pioneers in the field, and they all of them used electron microscopy in their research to study biological machinery, but they also really fostered the development of the field into what we know it today as a very cutting-edge technique. And they started doing this workshop that would happen at Scripps, and it became quite popular, where they would have people from all over the world come, different very esteemed guests like Professor Richard Henderson, who then went on to win the Nobel Prize for his contribution to a special type of electron microscopy called cryo-electron microscopy, as well as professors Bob Glaser and David Duracier, who also made seminal contributions to the field. And although Professor Duracier spent most of his career in Boston, we're very fortunate to have him be part of our La Jolla community here. And for an electron microscopist like myself, it's very special to have him even in the audience tonight. And so at Scripps, I was studying a part of the cell that we can think of as like the train and the train track of the cell, otherwise known as dynein as the train and microtubules as the train tracks. And what we knew about these is that they were very important for shuttling things around our cells, which you can see here. These are the green dots are the dynein zipping around on the train tracks of the microtubules. But we didn't have a complete picture of what they look like. And without a complete picture of what we look like, we don't really understand fully how they function. And so in graduate school, I used electron microscopy to transform these green dots into more detailed pictures of these molecular transporters. And in doing so, I made a very important discovery, which I'll show you right now. It was the fact that everyone up until this point had always thought that there was just two engines on this train, and I'm highlighting these engine components right here. But my work showed that there were indeed 4 engines that were hooked up onto this. And so this was a complete paradigm shift in the field for how we think about how this transporter works. And really, it sort of, at the molecular level, shifted our idea that maybe they're not like trains, but maybe more like supercharged chariots, kind of like these horse-drawn chariots in this famous action scene from Ben-Hur. And so this impact of this work was pretty profound on the field. And because of this, I was recognized as one of 13 recipients of the very prestigious Harold M. Weintraub Award. And I think I was one of 2 students to receive this in Scripps history. So this was a very proud moment for me that I was able to use this cutting-edge technique to make this new discovery. And this led to my graduation from my PhD advisor, Gabe Lander's lab, as the first student in his lab to graduate. And so at this time, as Jamie mentioned, I was sort of thinking about what I wanted to do next, and I was very excited about the idea of starting my own lab. And Scripps had just started the fellows program, so I joined as the 3rd fellow behind Michael Bolong and Michael Erb. And I knew I wanted to continue using cutting-edge microscopes to make really impactful discoveries. However, in the last few years before starting my lab, several things happened that really expanded my scientific motivation and made it a bit more personal. The first was that my grandma Pat was diagnosed with Parkinson's disease. And although this went relatively unnoticed and undetected for a while, especially when I was a graduate student. She started to decline quite rapidly. And then a few years later, my mom was diagnosed with a very aggressive form of brain cancer called the glioblastoma. And so while both of these diseases are incredibly devastating for both the patient and their loved ones, at the cellular level, they're very different. They both affect these cells within the brain, but Parkinson's disease is what's considered a neurodegenerative disease. So it's inherently characterized by a progressive loss of cells, whereas cancer is the opposite. It's characterized by an uncontrolled growth of cells within whatever region of the body, in this case, my mom's brain. But one unifying factor, despite being so different, are these tiny compartments that are within these cells called mitochondria. And so I realized that a key to my understanding these devastating diseases was to understand more about the mitochondria. So it was at this moment that I decided to dedicate my scientific life work to understanding more about how mitochondria function in healthy cells and what can go wrong in disease states. So you may not think that you know anything about mitochondria, but Jamie already gave away something very important. Mitochondria have the best catchphrase out of all the cellular components, right? Say it with me. Mitochondria are the powerhouse of the cell, right? And this is a pretty good catchphrase, I have to say. I mean, mitochondria do indeed generate energy for almost all cells in our body. But over the last few decades, we've really begun to appreciate in the field that mitochondria do so many other things in the cell. So many things, in fact, that I don't have time in this lecture to talk about all of them. But two I want to focus on are their roles in both powering cellular life through their role in energy production and metabolism, as well as their paradoxical or opposing role in triggering cell death through a pathway that is known as apoptosis. So you can see they have very important roles in dictating whether a cell lives or dies. And because of these important roles, it's probably not a surprise that in addition to neurodegenerative disease and cancer, mitochondrial dysfunction is associated with a myriad of other diseases. These are just a few of them listed up here. And so understanding what goes wrong in disease was really important to trying to think about ways that we could therapeutically target them. And although mitochondrial dysfunction is a hallmark of many of these diseases, in the field we've had relatively limited success in actually targeting the pathways that lead to mitochondrial dysfunction. And in my lab, we propose that this is in part because we don't understand an important part of how they function. And this has to do with how they're shaped. And if we knew more about how they were shaped, we might be able to address some of this in the field. So what is the right shape of mitochondria? Well, up until this point, I've showed you this, I've presented to you this very green egg-shaped looking entity and put a label as mitochondria there. So you may trust me that this is how they look. This is often how they're presented in the majority of high school and undergraduate biology textbooks. But the reality is, and if you take nothing else away from this lecture today, I want you to remember that the reality is mitochondria can take on many different shapes and sizes, and they're constantly in motion. And what you're looking at here is a very beautiful time-lapse movie that was generated by a postdoc in my lab, Nitish. And he's harnessing a technology that's similar to the phenomena. Has anyone ever seen the bioluminescent waves off the coastline? Yeah, so biologists and researchers, we've sort of harnessed aspects of that to enable us to illuminate parts of the cell that we want to look at. And so in this case, you're looking at a single cell here where all of the mitochondria are lit up. And we're looking at, we're taking images every 5 seconds. So I hope you can appreciate that, few, if not any, of these mitochondria look like an egg. And so hopefully I've convinced you that they look quite different than even how the textbooks present them. And the two main processes that dictate their shape are the processes of fusion, or the joining together of mitochondria. You'll see that happen here in this movie. And the opposing process is the process of fission, or the division of the mitochondria, which I think happens somewhere around there. And so these two fission and fusion processes are really what dictate the shape of the mitochondria, and they're really important for cellular and mitochondrial function. And if you were to just take a snapshot of the mitochondria in the cell, like I'm showing here, under normal conditions, you get what I think of as sort of this Goldilocks, what we call a network, all the mitochondria in the cell. And this is reflected by this balance of these opposing fission and fusion processes. So you have some big mitochondria, some large ones, and everything in between. But in some conditions where the environments are changing, you can have a hyperactivation of one of these two opposing processes. So for example, when there's a limitation of nutrients within the environment, oftentimes there will be an excess of the fusion pathway, which leads to mitochondria becoming super hyperconnected. It's kind of like, to me, they look like spaghetti or linguine all within the cell. And in general, this is associated, the shape of mitochondria is associated with more functional mitochondria that promote cell survival. However, some conditions within the cell can hyperactivate fission, which turns the linguine more into like a risotto. And this leads mitochondria to becoming more damaged or dysfunctional. And by and large, this is associated with cell death. And so while we understand and appreciate that mitochondria are dynamic, we're still missing a lot of information about how this happens and what it really means for the cell. And so this is something that my lab is really interested in studying. And so what we're doing is we're providing more detailed views of mitochondria, even more detailed than these images I'm showing you, to try to understand this. And we're harnessing technology, the same technology that's used for semiconductor technology. So every modern electronic has a semiconductor in it, and it's used and it's etched in this way using a similar device. We are using this device to etch into our cells and expose regions of the cell that have been previously unattainable by any other technique. And we can take those thin, newly exposed regions, and then we put them into our electron microscope. And then while in the electron microscope, we perform a microscopic CT or MRI scan. So if anyone has ever gotten one of these, you know that you sit in this tube and something kind of spins around you. This is kind of similar, except for this would be as if they were spinning you and the detector remained the same. We do a similar approach to generate all different views of our cells and the mitochondria within the cells. And then we use computational algorithms, fancy math, to take all of that information and reconstruct it into a three-dimensional representation of the mitochondria within the cellular environment, not unsimilar to a lot of the technology and algorithms that are used actually in the medical imaging community. And because Scripps Research is always at the forefront of cutting-edge technology, they were one of the first in the US to really make an investment in this technology that enables us to dive into cells and view mitochondria in this way. And in fact, when I started my lab in 2019, I was among one of the few researchers, the few dozen researchers in the world that had the expertise and the access to this technology. And so we really took advantage of this to look at mitochondria in different ways. And I'm just going to highlight for you some of the different ways that we're looking at mitochondria. So the first thing we wanted to ask is there anything different on the very detailed microscopic level between these linguine mitochondria and the risotto mitochondria? And in particular, we wanted to know something special about these structures that I've highlighted here, which are the barriers between the inside of the mitochondria and the rest of the environment. We call these barriers in biology membranes. And mitochondria are unique in that they have two membranes, which we call the outer membrane and the inner membrane. Now, while the outer membrane is quite smooth, the inner membrane forms this labyrinth of folds that we call cristae. And the shape of these cristae is really important because the shape enables them to function efficiently. And so we wanted to ask, is there anything different about these special folds between the linguine and the risotto mitochondria? And so here's an example of what our data looks like. To me, it's quite beautiful, and I can pick out the mitochondria in 5 seconds, but I appreciate that to most in the audience, it's just like a weird Jackson Pollock painting. So in order to make it a little bit more digestible to us, what we often do is we make these three-dimensional models that we call segmentations, where we only take out the piece that we care about, in this case, the membranes. And I'm showing the outer membrane in purple and the inner membrane in pink. And I hope now we're all becoming microscopists, observers, we can describe, I'll go first, what these cristae look like. And to us, what we thought they looked like were kind of like rungs on a ladder. You can almost imagine the temptation of wanting to just like scale up one of these as if you're scaling your roof to repair it. And so these were what these cristae structures looked in those elongated mitochondria, which represent the more functional pro-survival state. What about those risotto mitochondria, the fragmented ones? Well, to me, this is going to show in a second, the cristae looked more like sad balloon animals. They were very swollen and didn't look anything like the ladders that we had seen in those elongated mitochondria. And so we knew we were onto something because nobody had ever really looked at them in this detail. But if you've been paying attention, you know that we are not satisfied by talking about ladders or sad balloon animals. We want to transform these words into quantifications. And so inspired by a lot of the work I did as an undergrad, my lab dedicated quite a bit of time to figuring out how we could transform these models and these images of mitochondria into useful numbers so we could look for meaningful differences. And we actually borrowed some methods from the computer vision community. So anyone who drove here on autopilot mode in their Tesla, behind this is data that's used called LiDAR data, which represents the complex geometry of three-dimensional objects within the environment. We were inspired by similar technology to be able to represent our data in a completely new way that opened up the possibility for us to quantify it very precisely. And so what we did is we used this information to represent our membranes as a meshwork of triangles. And so this is very exciting to me, and maybe you now, because we love quantifications, when I tell you that each one of these triangles gives us an opportunity to make a measurement. So look at how many triangles. There are hundreds of thousands of triangles that we can use to measure and describe how these membranes are structured. And so we went crazy on this. We measured all sorts of things. We measured the distance between membranes, how their curvature, which is a fancy way of saying how sort of like curved or flat they were, as well as their orientation. And this is my favorite way that we represent our data, because to me, this is sort of where biology meets art, meets quantifications. What we're doing is we're essentially assigning a value, so in this case, the distance between membranes, to a color. And then each triangle that has a certain value encoded, a measurement encoded it, we're representing that value onto the membrane itself. So it's kind of like looking like a topological map of the US where you can see all the regions of the mountain chain. It's a little bit different analogy. It's certainly at a different scale, but this is a really powerful way to look for local changes in those quantifications. But of course, one of our main goals is to compare across the different groups. And so what I'm showing here is some of our data where each of these points represents a mitochondria that we've measured, and we're looking at the distance between that outer and inner membrane, and we're comparing between the elongated or the functional pro-survival and the damaged pro-cell death. And what we do in biology is we run our statistics, and then we represent that as asterisks here. And generally, the more asterisks you have, the more differences there are. And so what we found is that there were many differences, like we were seeing visual, but now on a quantitative way. And in fact, we took many different measurements. I don't have time to go into all of them, but what we found is that whether or not we were looking at these pro-survival linguini mitochondria or the risotto ones, there were significant but opposing changes at nearly everything we measured. And this is important to us because it suggests that if we can learn the shapes of these membranes at this detailed level, we can understand more about how mitochondria are functioning or not functioning in different states. And so this has really opened up a lot of opportunities for my lab to probe this further to understand what is mediating these changes. But of course, when you're the first to look at something, you have an opportunity to see things that nobody has ever described before, which is exciting and also anxiety-producing. And so it's kind of like being an archaeologist going on a dig, except for we like to think we're molecular archaeologists. And one of the things that my grad student, who's very talented and very observant, found were these dome structures in the mitochondria that we started calling volcanoes or rainbows. And so this is another really exciting opportunity that we have to now try to figure out where we have something. We have the ancient relic, but we don't know what it is. And so now we're doing all of the experiments to try to figure out what it is, but we think we're onto something because we think we see, although this is very preliminary, that how these are organized in the mitochondria changes depending on whether we're looking at these different linguini versus risotto mitochondria. Another exciting area that my lab is interested in is really probing what's happening during this fission process. Because as I mentioned, it's really important for mitochondria, but if it goes haywire, it's associated with dysfunction and many diseases. And so we were the first to apply this cutting-edge technology to look at mitochondria dividing. And you can see that here, this purple is this dumbbell-shaped structure is gonna be the mitochondria. And what we're interested in looking at are sort of these cables running across it, which kind of look like guitar strings to me. These cables are, as well as these blue structures that are kind of sandwiching the cables, they're really important for dictating where and when a mitochondria divides. They're kind of like the components that exert a little bit of peer pressure on the mitochondria to tell it when and where to divide, but we don't know how they do this. And so by capturing the detailed organization of all these components, we're trying to build a better picture of how they might be functioning in this role. Finally, the last thing that we're interested in is trying to think about how we might be able to turn the knobs on the system. And Jamie mentioned that my lab is considered one of these basic biologies or fundamental biology labs. But I have to tell you, being at Scripps and being surrounded by all of these drug discovery labs rubs off on you a little bit. And so I'm very fortunate to be able to partner with labs that are very good at doing this. And the lab I'm partnering with is the lab of Professor Luke Wiseman. And we're trying to figure out if we can reverse or prevent this fragmentation because it can be very destructive. And so through a collaborator, we've gained access to cells that were graciously donated to us by patients who have mutations that these mutations lead them to have mitochondria that are very fragmented. And so we were curious if we could actually identify molecules that would turn these risotto mitochondria into linguini. And so we identified 2 molecules that look promising to do this. Of course, no one in the audience is satisfied just by these images. My grad student wasn't either, and so she developed a nice approach to be able to quantify these. But we really think that this is the beginning of something very exciting. Now, I want to caution, we are very far, far away from any sort of idea that these could be used as treatments or medicines or therapeutics. Really what we're excited about is the potential of using this as a tool to understand mitochondria more. However, the more that we use this tool, the more that we can help design and understand this system to one day potentially lead to new therapeutics for some of these disorders associated with mitochondrial dysfunction. And so I hope today what you've taken away from this lecture is that mitochondria are not just the powerhouse of the cell, they're actually much cooler than that. They take on lots of different shapes inside of the cell, and that these shapes are more and more we're starting to appreciate important for their function. I hope you've also gained a little bit more of an appreciation, like me, for the power in using microscopes to really visualize the unseen world, and especially an appreciation for how much of life is important beyond what we can see with our human eye. And similar to how life extends across scales, I want to acknowledge all of the support I've received across different scales. So first, I want to acknowledge my incredible team of researchers here, both past and present. I just want to say what an honor it is to be part of this team. You guys are so talented, and I'm trying to be serious while this video is going. But yeah, in all seriousness, you make it a joy to come to work every day and work with you, and I couldn't have done it without you. I also want to acknowledge the support of my friends and family and colleagues, some of which are in the audience today. I want to acknowledge my 3 best girlfriends who have supported me throughout my journey, as well as my colleagues, and in particular my husband, Michael, who uprooted his life to move 2,000 miles away so I could go after my microscopy dream. So thank you so much. And finally, I want to acknowledge the community's support. I've been very fortunate to receive financial support from several foundations that really enable me to go after these bold and brave scientific ideas. And lastly, I want to thank all of you, both in person here tonight and online. Thank you for taking time out of your busy schedules on November 6, 2024 to come here. And whether you are like Sheila Wiseman, who have seen every front row lecture, or this is your first one, thank you so much. It's been such a pleasure to be here, and I look forward to the Q&A. Now more than ever, we as scientists really need the support of the community. And I think this support is 2 ways. And I view my role as a scientist and talking to the community as really important to convince you all that what we're doing is important for human health. And so thank you so much for coming on this journey with me, learning about the 2 very important women in my life and how this has transformed my view of science. And I also want to say, if you're interested in participating more in this journey, there's many ways that you can contribute to the scientific process. Of course, the most direct way is through a financial investment, but if that's not really in the cards for you, I want to encourage you to really think about how you can be an advocate for scientific research in your own communities. I think it's pretty simple because pretty much every medicine that you or someone you love has taken came from a discovery like labs like mine. And so talk to your friends, neighbors, and colleagues about the cool science you've heard at Scripps Research and really advocate for policies that will promote scientific funding. And continue coming to these wonderful events. We love seeing all your faces, and I look forward to the discussion. Thank you so much.
I think we should take you on the road. Great job. So if someone has a question, put your hand up and we'll get to you with a lightsaber. There's someone with that. I'm still thinking about the fusions and the fissions. And then you talked about how, there's an outer membrane and then there's the inner membranes. And so if you're pinching off, you got to pinch 2 of those things and make sure they don't get confused. And then if you're joining, you have to break both of those open.
So how does it all work?
It's just, so it's a lot. It's more complicated than just a cell dividing where you get one membrane. So
absolutely.
So how does that all get organized?
Yeah, I mean, you've touched on a big question that we have in the mitochondria biology field. And I sort of touched on it here a little bit in my talk, and the reason why we still have it kind of seems like a fundamental question. How do you have 2 barriers that need to divide? How do you do that? But really the challenge is that while we're able to see what can assemble on the mitochondria just before it divides, we don't have a detailed picture of what's actually happening to those membranes when it divides. This is something that my lab is really interested in discovering. And what we think is happening is that it depends on the context. It depends on, and I didn't go into this, but there are many different pathways to how this fission, this division can work. And so that opens up a whole box of complexities. So the short unsatisfying answer to your question is we don't know.
That's,
but it leaves a
that's
lot
why
of
we hired
exciting.
you.
Yes.
Yeah. So maybe the other thing, just in general, I found myself wondering about, what's the time scale of it? So you you have linguini, you have risotto, you have normal, and so you get up in the morning and you're hungry. Are you in linguini land, or what's, for a normal cell, what is the time scale under which you would undergo these morphological changes?
Yeah,
is it like all the time, or is it, you have to be really hungry for a long time before you get this?
That's a great question. So it's difficult to say about every mitochondria and every cell in our body. But there is some interesting research that suggests that this fusion and fission process is connected to our circadian rhythm, right? Our light and day cycles, what tells us when we should go to sleep at night when it's dark and when we should wake up in the morning. For us, in a laboratory setting, we also often use model systems. And what that means is that we have sort of a simplified version of what might be going on inside of our body. And oftentimes we use little tricks to help us tip it one way or the other. And so we have very powerful tricks that we can actually just put a small molecule on, and within seconds, the mitochondria divide. And so this is how we're able to kind of push the balance and study these complex processes. So I think there's still a lot more to know about what's going on there, but again, gives us lots of areas to cover.
Great. I see we have a question back here. Please.
Yes, thank you. You've talked about changes in morphology of the mitochondria in sort of healthier disease situations, but I was wondering about the morphology in different cell types, and if you can learn anything about how any differences in cell type relate to like energy consumption or other factors that might go into this.
Yeah, that's a great question. If I had, hours with all you, I could have gone into even more of the complexity. You hit it right on the nail that there are, the way mitochondria are shaped, I presented a somewhat complex but simplified view of this, in that the reality is that it really does depend on what tissue type you're looking at. And what's cool about the mitochondria is even though they have all of the mitochondria in our cells in the human body have the same basic makeup, the shape can be fine-tuned for its function. So for example, the mitochondria in the heart, they are supercharged to be energy producers because that's really important for the heart, right? And so their shape and their structure is very different from, let's say, for example, the mitochondria that I was showing here, which are more like reminiscent of mitochondria that might be within the cells of our skin. And so there is a lot to learn about the shape of mitochondria in different tissues, and this is something that's important for us as researchers to take into account, that when we're looking for changes in shape, we need to account for that what we're comparing to is really apples to apples and not apples to oranges, or brain to kidney, so to speak.
Oh, question down front. Yep.
That was a fantastic talk.
Thank you.
Really
David.
wonderful. I started off as a physicist and then a biophysicist, so my view of a cell was a nucleus and one mitochondria. And I'm looking at the jillion mitochondria, and I'm wondering, how does the cell regulate how many of those things, what density it needs? I mean, surely there must be, they can't go wild dividing, so
Right.
there must be some way of regulating. It's a little outside what you're working on, but it just struck me as phenomenal how many there are.
Yeah, how does the cell keep track or keep tabs on all of them?
Exactly.
Yeah, again, it's very complex. There's lots of pathways that not only control what I talked about today, the division or the fusion of them, which really doesn't change the amount, right? You're just sort of breaking it up into pieces or putting it together like Play-Doh. But there are biogenesis, or there are pathways that lead to the creation of mitochondria when necessary. Something I think is cool is that the division of the mitochondria is coupled to the division of the cell. And when you think of this from like a practical perspective, it's like the cell wants to put the mitochondria into little pieces because it's easier to partition. It's like, I don't know, like partitioning things up. I don't have a good analogy on the fly for this one, but yeah, it's kind of like breaking something up into little pieces. It's easier to make sure they're evenly distributed, and I love the beauty of the elegance of that system.
We have a question back here.
Hi, thanks for the talk. I'm curious about kind of the transfer of like healthy versus diseased mitochondria between cells. You spoke a bit about pharmacological methods to kind of turn, the linguine into risotto, or vice versa. What about like, tunneling nanotubules to take healthy mitochondria and transfer them into, diseased cells?
Yeah, this is an area that people are looking into, this idea of can we repopulate damaged mitochondria populations with healthy ones. This is something that my lab isn't doing, but is certainly something that's in the field. And one thing I'll say about that is actually this is a strategy that some cancer cells can use to actually proliferate. And in particular, the type of cancer my mom had, it's known that those cells are actually taking mitochondria from other cells to really enable them to continue to proliferate and divide. So it's kind of like pirates, like hijacking them. So this, in terms of a therapeutic, I think there's challenges, as you can imagine, in doing this. But it's definitely a strategy that people are pursuing.
Thank you.
So one, I'll come to you just in 1 second. Danielle and I actually collaborate.
We do.
We have a joint project. And the one thing I can confirm for the audience is every once in a while, we get to a certain point and Danielle just jumps up and starts making lists on the blackboard. So this part, I've seen this a few times. So my question is, and in some of the pictures, there was all these big black dots all over there, which are in fact the ribosomes.
Yes.
But
I'm sorry I didn't talk about the
ribosomes.
I'm sorry. I know, I'm sorry.
I'm not feeling it. So, but it made me think about, so there's plenty of ribosomes that are on the outside of the mitochondria.
Yep.
And they're making proteins that go inside the mitochondria.
Sure.
But so I never really thought about it, but you're fusing and fissioning, and but it's not only ribosomes. There's all this stuff out there. So how do you choose where do you clear the
where they go?
Just clear the counter and then divide, or
yeah.
What is going on?
Yeah, so I can indulge and talk about ribosomes because they're related to the mitochondria. So yeah, what's interesting about the mitochondria is that, Jamie mentioned this, I didn't talk about it at all, but they're unique in that they contain their own small genome, or DNA. So this is separate DNA from that we think about is in the nucleus. And this is sort of an ancestral relic of how we think mitochondria came to be, which is that there was a bacteria that engulfed another bacteria and then retained some of that DNA. But what's interesting is even though they have their own DNA, it only encodes for 13 proteins, and there are over 1,500 proteins that make the mitochondria function. So there's clearly a lot of cross-communication. Proteins have to come in, they go out. The ribosomes, there's a small set of proteins where the ribosomes actually dock directly on the membrane itself. This is a very rare and elusive process that my lab is working on. We have a paper in review right now looking at the structure of these. But something else that's interesting is mitochondria have their own ribosomes. And so whether this idea of ribosomes on the outside coordinating with ribosomes on the inside is something that people haven't been able to really test before, and something that we're interested in looking in.
Okay. Here into the ribosome discussion. Thank you. I appreciate it. Down here, question.
Great talk, Danielle.
Thank you.
I'm curious, you talk about healthy cells and cell death. The question is, does the disintegrating mitochondria cause cell death, or is it a symptom of cell death?
Yeah, that's a great question. So mitochondria are a really important part of what we call the cell death pathway, or apoptosis. And there's a molecule that's actually normally kept tightly within the mitochondria, within those cristae, called cytochrome C. And it's only through a series of very regulated events that that molecule gets released into the cell. And there's a reason why it's so regulated, is because once that molecule is released, it really triggers this cell death pathway. So you don't want that to happen willy-nilly to any cell types. But there are indirect, so that's a direct way that mitochondria contributes to cell death. But there are indirect ways, right? If the mitochondria aren't able to produce enough energy for the cell, this is going to mean that there's a lot of downstream effects where there's not enough energy, there's not enough proteins being made, they're not functional. And so that's sort of like an indirect way that the dysfunction of mitochondria may contribute to triggering the cell death pathway, although it may not happen through the mitochondria itself.
Okay, one more up here.
Hello, great talk. I was curious, the mitochondria can reside in multiple positions within one cell. Let's say, for example, a projection neuron can be very lengthy. And in that case, do you think mitochondria status is synchronized across the cell, or it is more like a local manipulation?
Yeah, I think your question is getting at when you have these specialized cell types that have very long projections, is there something unique about those mitochondria? And I think we believe that there is something unique about the mitochondria that are close near the nucleus of the cell, which is a lot of the mitochondria I showed today, versus those that are farther out in the projections. And they might actually have different roles, whereas some of them might be more involved in producing energy locally in those distal locations. Maybe the ones in the nucleus are performing other functions. And so, yeah, and certainly there are differences. And actually, we have a really nice collaboration with a lab here in the neuroscience department, really trying to look at how the structure of mitochondria changes, whether we're exciting or inhibiting a neuron. And so stay tuned for more on that.
All right, I think we're at the hour. I just maybe ask you one last kind of just sort of, future forward-looking statement. So you brought this cryo-ET and the fib milling and all of that to Scripps, and so cutting edge puts you in the position to ask questions that other people can't. But, so what's the next great thing? Where are we going in structural biology so if I had a checkbook, what would be the gadget that I could buy you that would,
yeah.
So where are we
well,
going?
I got a list, so
Okay.
I only can choose one? Okay.
It's always good to have a list.
Well, okay, so the cutting edge technology when I started my lab was I showed it in that animation where we're micro-machining individual cells. And we're doing this to expose regions. But of course, the dream is to be able to do this on more complex systems like tissue, right? We could look, for example, at an entire zebrafish or an entire small worm that is often used in research. And it becomes more complex as you have something that's a little bit bigger to do this for technical reasons. But there are technologies out there now that would enable us to look at that. And the reason why that matters is because that would represent more the physiological state, right? Ultimately, when we're removing neurons or cells from a body, we try our best to recapitulate what it's like when they're inside of our body, but there are caveats to that. So being able to do it within its native environment is really, I think, the holy grail. And so that's the item I would put on my wish
all right.
list.
Coming right up. So, all right, well, look, thanks. So I hope you'll agree that the future of Scripps is secure. We're really proud to have you here as a faculty member. And unfortunately, this is our last front row for the year. We'll take a break. Stay tuned. There will be another calendar year, but I think we're scheduling, and the next one will be in February. So thank you all. Have a great holiday period, and welcome back to the front row. Great.